cellulose is about 26 to 43%, lignin about 21 to 31%, and pentosan about 15 to 25%. However, ash about 1.75% only, and silica about 0.7%. So these are the chemical composition of bamboo. So let's see how this chemical composition travel into the structures that explained by Zafira. So we'll proceed with Ain.
Okay, we proceed with the process of digestion in the buffalo. Buffalo are like cattle, ruminants. This means that they can utilize microorganisms in the rumen to digest the feed. The feed eaten by the ruminants are mainly of vegetable origins. The ruminant is an expert in converting cellulose and other fibrous material into high quality meal and meat. Their digestive capacity is greater than the non-ruminant. When ruminant eats, it does not chew the grass but swallow it immediately and store it until its appetite is satisfied. Then the food is returned to the mouth and the buffalo chew the cuts. When the food is first swallowed, it passes down the esophagus into the first chamber called the rumen. In the rumen, bacteria and protozoa secrete the enzyme cellulose to hydrolyze the cellulose into glucose. The food passes into the second chamber called reticulum as the rumen is full. The bacteria and protozoa in the reticulum continue to hydrolyze the cellulose. When the ruminant is ready to chew the cud, the food is returned to the mouth and to be chewed again. Movement of the food from the reticulum by the antiperodosis of the esophagus. After the food has been chewed, it is again swallowed, but this time it passes into the third chamber called O muscle. The inner surface of the O muscle has food to act as seal. Because the product of cellulose digestion is absorbed, and as the food passes into the last chamber of abo muscle is trapped. This is to prevent any big particle from entering the abo muscle and damaging it. The abo muscle is the true stomach and it is the only chamber which secretes gastric juice for digestion of other food substances. The digestion in ruminants is a good example of symbiosis. Microorganisms present in the stomach of ruminants help in digesting cellulose and obtain shelter and nourishment from the animal. The symbiosis relationship exists between the microorganism and the ruminants. That's all about the process of digestion in the buffalo.
Maybe you can do and draw the sequence and the photo.
Yes. So the what was it? So in the digestion of human or buffalo or cattle, we have the absorption of nutrients. So we use volatile fatty acid. One of them is volatile fatty acid. So what is volatile fatty acid? Volatile fatty acid is actually also known as short-chain fatty acids that is produced in gastrointestinal tract by microbial fermentation of carbohydrates and also indigenous substrates such as mucus. So this can be of great advantage to the animal, since no digestive system, no digestive enzyme exists for breaking down cellulose or other complex carbohydrates. So VFA is used to break down cellulose in animals. And also the VFA or volatile fatty acid are produced in the largest amount in the herbivorous animals such as cattle, and especially in the forestomach of ruminants. VFA, however, also are produced in the lower digestive tract of humans and all animal species, and intestinal fermentation resembles that occurring in the rumen. So we also have volatile fatty acid. The principle, everything in this whole process is so does VFA. So the principle of VFA in either the rumen or large intestine are acetate, propionate, and also butyrate. These three principles I will explain later. So that is produced in the ratio. The produced in the ratio varying from approximately, first ratio is 75 to 50 to 10, to 40 to 40 and to 20. So there are two ratios. Absorption of VFA at their site of production is rapid, and large quantities are metabolized by the ruminant or large intestinal epithelium before reaching the ruminant or large intestinal epithelium before reaching the bottom line. Most of the butyrate is converted to ketone bodies or carbon dioxide by the epithelial cells, and nearly all the remainder is removed by the liver. Propionate is similarly removed by the liver, but is largely converted to glucose. Although species differences exist, acetate is used principally by peripheral tissues, especially fat and muscle. Considerable energy is obtained from VFA in herbivorous species, and far more research has been conducted on ruminants than on other species. Significantly, VFA, however, are now known to be produced in omnivorous species such as pigs and also humans. Current estimates are that VFA contribute approximately 70% to the caloric requirements of ruminants such as sheep, cattle, and mushroom. Approximately 10% for humans, and approximately 20 to 30% for several other omnivorous and or herbivorous animals. The amount of fiber in the diet undoubtedly affects the amount of VFA produced, and thus the contribution of VFA to the energy needs of the body could become considerably greater as the dietary fiber increases. Pigs and some species of monkey most closely resemble humans, and current research should be directed toward examining the fermentation processes and VFA metabolism in those species. In addition to the energetic or nutritional contributions of VFA to the body, the VFA may indirectly influence cholesterol synthesis and even help regulate insulin or glucagon distribution. In addition also, VFA production and absorption have a very significant effect on epithelial cell growth, blood flow, and the normal secretory of absorptive functions of the large intestine, cecum, and also rumen. The absorption of volatile fatty acid and sodium, for example, seem to be interdependent, and release of bicarbonate usually occurs during VFA absorption. So the three principles is the first one. The first one is acetic acid. So acetic acid is 50 to 60% of volatile fatty acid produced is acetic acid. It predominates on a high fish diet and is a precursor for mammalian fat. Some is also used for muscle metabolism and body fat. The molecular formula for acetic acid is CH3COOH. For propanoic acid, 12 to 18% of VFA produced is propanoic acid. Predominates on a high concentrate diet and provides energy via the conversion of blood glucose into the liver. It is used in lactose or milk sugar synthesis, and the molecular formula is CH3, CH2, and COOH. The last principle is butyric acid. 18 to 20% VFA is produced is butyric acid. Provides energy to the rumen wall and used in milk fat synthesis and for body fat. When excess energy is present in the diet, it doesn't vary in proportion to other volatile fatty acids. Therefore, has little influence in milk fat content. So the molecular formula for butyric acid is CH3, CH2, CH2, COOH. So a lot of us clear that.
So now I will talk about the process takes place in rumen more in detail. The feed will enter the rumen compartment and swallowed by the buffalo. The rumen is the largest compartment, as mentioned by Zakira just now, with a volume of 150 to 200 liters, or in other words, 40 to 50 gallons. It is an anaerobic environment in which no oxygen is present. The feed is exposed to microbes such as bacteria, protozoa, and fungi. They help the cow to digest and utilize nutrients in the feed. These microbes attack the feed particles, and by enzymatic action, the components are broken down and used for their own metabolism, growth, and reproduction. The feed is massively feed, masticated, regurgitated, and exposed to microbes in the rumen. Large particles will become smaller and eventually be transported to the reticulum and further on. How long time a specific feed particle will stay in the rumen depends on size, palatability, and deep fiber content of the feed. The buffalo has a slower rumen movement than cattle, which leads to a slower rate of ingest outflow. To achieve efficient feed utilization, the reticulum must have optimal conditions. pH of the rumen content is similar to that of cattle, and it is affected in the same manner. Normal pH is between 6 and 7 depending on feed and time of feeding. The process of fermentation takes place in the rumen where microorganisms convert carbohydrates into volatile fatty acids and gases. This process allows the cow to convert cellulosic fiber into energy. Of gases produced within the rumen during fermentation, which are 500 to 1,500 liters per day, or in other words, 150 to 400 gallons, 20 to 30% consists of methane and carbon dioxide. Production of fermentation gases represent a considerable energy loss. Certain fermentation modifiers, such as ionophores, improve energy efficiency of ruminants by reducing those gas energy losses. The fermentation gases are expelled by reduction. When reduction is impossible or ineffective, cows can suffer from bloat. Volatile fatty acids, VFA, together with ammonia, are absorbed by the animal through the rumen wall and transported via the blood to the liver where they serve as building material for chemical compounds such as glucose, protein, and fat. Ammonia can be utilized directly by the rumen microbes to synthesize proteins. To be correct, one is actually not feeding the buffalo, but its microbes. Ruminants are entirely dependent on the function of the rumen microbes. Therefore, it's important to keep the rumen environment healthy. The easiest and best way to feed a high amount of good quality roughage and a smaller amount of good quality concentrate. That's all for me.
Before making a conclusion, I would like to talk about the learning outcome to conclude what I said just now. The objectives of this activity is to know about the effect of excessive consumption of bamboo in buffalo, or also known as target fermenter, which is the answer is protein. Protein. And not only that, figure out probable biochemical events and processes that took place during the digestion of bamboo in buffalo, that which I just explained. And moreover, to examine the role played by nutrients in controlling feed intake in buffalo ruminants. The benefits of taking good nutrients and effects arise when there is lack of nutrients. To know the effects of volatile infusion of fatty acids in feed intake in ruminants. Apart from that, to know the chemical composition of bamboo as well. Finally, to figure out the scientific terminologies to describe the digestion process in buffaloes. I believe that we have covered that all the objectives that we wanted to explain. And right now, I would like to explain several words that is several scientific words that we don't really understand. The first thing is cellulose. You guys know what is cellulose? No. Cellulose is a combination of cellulose and hemicellulose, and usually accounts for 65 to 70% of the dry weight. And the second one is bloating, which is swelling, and the more precise explanation is over distension of the ruminal reticulum with the gases fermentation, either in the form of persistent foam mixed with the ruminal contents called primarily of frothy blood, or in the form of free gases separated from the ingest called secondary or free gas blood, which is protein aspect of stomachs. Thirdly is hemicellulose. Hemicellulose is any of several heteropolymers such as arabinolases present along with cellulase in almost all plants or walls.